Hello everyone, hi, welcome. This is a video to look at the review of the recent cells test that you had a go at. Alright, so we're going to go through the questions, we're going to look at the answers and also talk about some kind of exam technique that will help you to try and improve. So this will obviously cover eukaryotic, prokaryotic cells. It will also look at cell microscopy. We'll be looking at mitosis as well within that, and also looking at cell magnification and cell size. I hope you enjoy the video, and it'd be great to hear your comments on it as well. Okay, so let's get started. Okay, on to the first question. This question is what we call an AO1 question, so it's knowledge and recall. So as you can see, first of all, it says to describe how you could use cell fractionation to isolate chloroplasts from the leaf tissue. So the key thing with this question is that they want obviously you to talk about cell fractionation, but they then want you to go on to say how you would isolate chloroplasts from the leaf tissue. Okay, so the first mark, you've got to mention that you would cut up the tissue and you would break it up with a homogenizer possibly, so broken up with homogenizer, and then also, and I'll put that in capital letters, remove the cell debris to get just one mark, okay? For the second mark, you need to mention that the solution is cold, or you could mention isotonic or buffered, and so that's just for one mark, okay? And then it asks how you'd isolate chloroplasts. Well, basically you'd need to centrifuge, and the second pellet, you need to mention the second pellet is the chloroplast. So there are the answers, okay, and you can see you've got to be quite specific. I just want to note that for the second mark you need to just mention either cold, isotonic or buffered, not all three, you just need one of those. But for the third mark you do need to say that if you were to use a centrifuge that it would be the second pellet, which is the chloroplast, because that answers the question. Okay, on to 1B. Again, it is an assessment objective 1, so knowledge and recall, and it just wants you to name the parts of the chloroplast labelled A and B. So as you can see, the answers A is stroma, B is granum. They would accept thylakoid as well for that answer. Okay, 1C, as you can see, you need to calculate the length of the chloroplast shown in the figure above. So the first thing you need to do is know what you are going to do with your calculation, and you're going to use the I am triangle. Ta-da! So there we go. Okay, so we can now look at what we need to do. So the length of the chloroplast, you're going to need to measure the image, and you're going to measure it in millimeters. And then the thing is, you need to know the length of the bar as well. So if you look at your diagram, you need to measure that in millimeters, okay, and then you need to look at the length of the chloroplast divided by the length of the bar. So in my measurements, I measured the chloroplast length as 72 millimeters, and looking at the basically 1 micrometer is 17 millimeters in my calculation. So what you do is you take 72, divide by 17, and you get 4.2. I need to check that answer 4.2 micrometers with my colleagues, but I think 4.2 is fine. Okay, 1D. Asks you to name 2 structures in a eukaryotic cell that cannot be identified using an optical microscope. So answers would include things, basically organelles such as mitochondrion, ribosomes, endoplasmic reticulum, either rough or smooth, lysosomes, cell surface membrane. Okay, so you need 2 of those to get the mark. Okay, the next question, question 2, is looking at a Chlorobacterium. So we're talking about prokaryotic cells here. It gives you some information. 2A, it says name A. And these are always a bit difficult. You've got to really look at the line very, very carefully. Okay, so looking at the line going A, it's going to the interior wall. So that will be a membrane, basically, and it will be the cell or the plasma membrane. 2B is basically asking for a bit of recall, but it's also asking you to apply your knowledge in terms of knowing the differences between eukaryotic and prokaryotic cells. So for this answer, there are quite a lot of answers to go through with this, but you only need 2 of the following. Okay, so if we look at the answers, way, look at that, that's good. Right, anyway, so 2B, as you can see, you need to mention that the differences between them. So, for example, in a eukaryotic, because it's talking about the differences between eukaryotic and prokaryotic, you can say membrane-bound organelles, but I mentioned that the nucleus is in a membrane. It contains mitochondria, and you've got ER, lysosome. Also, as it's a small intestine, you can mention the microvilli or microvillus. You've got to mention microvillus, not villi, okay? The Golgi body, you can mention linear or non-circular DNA. When you're mentioning ribosomes, look at that answer. It wants you ATS or denser or heavier or larger ribosomes. If you just put ribosomes, that is not sufficient, okay? So just watch for that. You need to give detailed answer. Okay, so 2C, it's talking about the Chlorobacteria again, so using TEM or SEM, okay, so again this is an A01 question, so it's recall. If you've done your homework, if you've done your revision, you should know the advantages of using each type of microscope. So, give one advantage of using a TEM rather than SEM to look at Chlorobacteria, obviously TEM has a higher resolution. You can say higher magnification or a higher detail, okay? Or you can mention that it allows you to look at the internal details of the structures within the cell. Answers that would be rejected are things if you say longer wavelength, for example, which is incorrect, or if you said it can be used on living specimens, which obviously can't because you need to use very thin sections. So on to C2, give one advantage of using an SEM rather than a TEM. Well, first of all, you don't have to prepare thin sections, okay? The SEM shows the surface in great detail, which is an advantage. But the main advantage I think that most of you will get is that you can have a 3D image. Again, answers that say that you can look at living specimens is incorrect, as the specimen will be placed into a vacuum. And as we all know, you can't have a living specimen in a vacuum, unless it's in a spacesuit. Okay, 2D, another cell microscopy question. So they now want you, for this question, calculate the actual width of the Chlorobacterium, and they quite conveniently and quite nicely give you points B and C to measure. And it says give your answers in micrometers and show your working. Okay, so to work out this answer, I've got my IAM pyramid here, and I think the first thing you're going to do is measure B to C. So B to C I measured, and I got, it might be incorrect, but here we go, well I got 19 millimeters. Okay, so 19 millimeters, and that's between, sorry, not A and B, B and C. And then it does say in the question, in the first part of the stem of the question, it tells you the magnification, and the magnification is basically, it is 50,000, so 50,000, okay. Okay, so going to my triangle, I know that the image is 19 millimeters, and I know the magnification. If I just put the magnification in, I know that it is, okay, it's 50,000. So I take the 19 millimeters divided by 50,000, and that gives me a really big number, 0.00038. But if you look at the question, okay, the question does say that it wants you to work it out into, get the arrow working, into micrometers. So your answer, you have basically got to times it by 1,000. So you always times by 1,000, all right, if you want to work out micrometers. And in this case, in my answer, I've got 0.38. So my answer is 0.38. I need to again just if you've got the right idea, I've probably given you the correct answer. In the real exam, they will give you a range. For example, if you look at the mark scheme, it actually says 0.42 to 0.46. The reason it's not the same is because the image has been slightly reduced on your test sheet. But it's always worthwhile showing you working. So even if you get the answer wrong, at least you get one of the marks. Okay, question 3, it shows you the diagram showing 4 stages of mitosis. It wants you to name stage A. Stage A is anaphase. The next question wants you to give the stages A to D in the correct order, so that would be starting off with C, and then it would be B, A, and D. So A3, describe and explain the appearance of one of the chromosomes. Okay, so key thing is there, describe and explain one of the chromosomes in stage B. Well, stage B looks like metaphase, so to describe the chromosome or the DNA has been replicated, so that's a description, and an explanation is that basically each chromosome consists of 2 chromatids, or you could have chromatids attached at the centromere. So 3B, they give you some interesting information related to using a substance called colchicine. Now, you wouldn't have learnt about that, okay, so this is obviously related to AO2 skills, so you're applying your knowledge in an unfamiliar context. The first question asks you what has happened to the chromosome number. So if you look back to the original picture, you'll notice that actually where there were 4 chromosomes, there are now, if you count them, you look on there and you can count them, and there are actually 8 chromosomes, which means that they have doubled. Okay, so you can either have 8 or you can have doubled there. Okay. So looking at the next question, it has this command word suggest. Now suggest is an interesting thing because there are several answers you can include. So you need to suggest why it has doubled. Well, basically the DNA has replicated. Definitely it's replicated because it's gone from 4 to 8, okay? Alright, but there's been no separation, okay? And the no separation, or you can say that there's no cell division, okay? So there's no cell division there. No cell division. Okay. On to question 4, and it talks about an amoeba, and it tells you that it's a eukaryotic organism, and they say that they use a TEM. Okay, so using a TEM to study the diagram. First questions, really good, okay, in terms of their AO1. So name the organelle Y. Organelle Y is the Golgi, either the apparatus or the body. Okay, and then so the A2, name 2 other structures. There are lots of different structures. For example, nucleus. You could say that about nuclear membrane or nucleolus or nuclear envelope. Mitochondrion, they'll accept mitochondrial membranes or cristae. endoplasmic reticulum, and lysosome. Although be careful with your spelling because they will reject lysozyme. 4b asks what is the function of organelle Z. Okay, so organelle Z, you have to just double check, and you know that organelle Z is a mitochondrion, but that's not going to get you a mark because you need to say what the function is. The function is respiration, or you could have ATP production, or it provides energy. Now there's a key thing here that you cannot say things like produces energy, all right? It doesn't produce energy, it provides energy, because it provides energy from the breakdown of ATP. On to 4C now. They mention the transmission electron microscope to study the structure of the amoeba. It says explain why. So it doesn't want a description of the TEM, but it does want you to say, well, why is it are they able to study the structure? And it's mainly about, basically, there is a better resolution of the image. And why is there a better resolution? It's due to the shorter wavelength of electrons. Okay, that's a stock answer. They ask that a lot. I would learn that kind of answer. You could also include, I know it's only 2 marks, but if you put one of those, and to see the internal structures or look at the organelles, examine the organelles, that would be acceptable too. So, question 5. The first thing to note about question 5 is it's 5 marks. So they're looking for 5 points here. So we need to look at it carefully. So it says that the structure of Chlorobacterium, so prokaryotic, is different from the structure of an epithelial cell in the small intestine. And it says describe, which is great because that's just recall of your knowledge. The structure of a Chlorobacterium is different. Okay, so you need to talk about the bacteria. Okay, not the small intestine, you're going to talk about the bacteria here and how it is different. And there are quite a lot of different answers you can get here for 5 marks. Well, the first thing you can say that it's not mentioned in the stem of the question. This is the stem. Okay, so if it's not mentioned in the stem of the question, you can mention it. The first thing is that it's a prokaryote. So that's a difference. Okay, so it's prokaryotic. And then in terms of the structures, you can say that basically no nucleus. That's one of the points. Okay, all right. Or it has, or you can say a loop of DNA, which is the plasmid. Okay, you could mention that there are no membrane-bound organelles. No mitochondria. You could also mention within that no Golgi, things like that, but you'll only get a maximum amount of marks for those. You've got to mention that they've got small ribosomes. Okay, so remember 70S ribosomes. Okay, and then any 2 from the following. Alright, you need to mention capsule or flagellum, okay, or plasmid, as I've already said that, and cell wall. Okay, cell wall, because that's different to the small intestine. So you can see there's lots of different answers in there. That is a really nice question. That is a good old AO1 5-mark question. If you've revised, you'll know those. Okay, question 6 is related to mitosis, and it's about looking at onion root tips. And this is doing if you remember, we did the squash, the temporary mount of the onion tissue on a glass slide with a cover slip, and it asks this. Okay, this is the question for 2 marks. It says, push down hard on the cover slip, but do not push the cover slip sideways. Explain why she was given this instruction. Well, we can split it up, okay, into 2 things. So, first of all, push down hard on the cover slip. That's the first point. So, I'm going to mention that pushing hard what that does is spreads the tissue, doesn't it? That's what we're doing, spread or squash the tissue. Spread or squash tissue. So that's for the first mark, and then we've got to say, well, why do you not do the cover slip sideways? Well, what the reason is that you're avoiding the cells rolling together. Okay. The problem with that is that you won't then be able to distinguish the chromosomes. Okay. So avoid the cells rolling together or breaking of chromosomes, because you're trying to observe chromosomes in mitosis, so you don't want to affect your view. Okay, this question, it shows you an image. Okay, that to me looks like anaphase, although it doesn't really matter, because we need to discuss, okay, 2 reasons for your answer. Okay, so looking at the diagram, you can clearly see that the chromatids, these things, okay, they are basically at the poles of the spindles, or the ends of the spindles, okay, not the cell, the ends of the spindles. You can just make out that they're at the end of the spindle. Alright, so that would be the first thing about chromatids, okay, or chromosomes, it doesn't matter, but chromatids at the poles of spindle, or the end of spindle. Okay, so that's the first point. Okay. And you can see that they're actually, if you look closely at that, they are actually a V shape, okay? And it does show, therefore, that the sister chromatids have been, or the chromatids have been pulled apart. So that's another reason related to anaphase, the chromatids pulled apart. But where have they been pulled apart? Well, they've been pulled apart at the centromere, okay? Okay, hopefully you can read my writing. And that's kind of the 2 questions. You don't get any mark for saying yes or no, it is or it isn't, but you've got to give 2 reasons for your answer. Those answers, that would suggest to me that that is anaphase. Okay, and now on to the last question. So we've got a bit of maths here. Okay, so this is a bit of apparatus and techniques. All right, so let's have a quick look. So this type of question, you need to really look at it carefully. Okay, so it tells you this person observed in each stage of mitosis, they looked at a total of 200 cells, and only 6 were in anaphase. And then it gives you some more information saying 1 cell cycle of onion root tissue takes 16 hours. Then it says calculate how many minutes, so not hours, okay, not hours but minutes, these cells spend in anaphase. Okay, so well, first of all, you've got to work out the proportion of cells that are in anaphase at that particular time. So that would be 600 divided by 200, and then you've got to convert 16 hours into 16 hours, sorry, into minutes. So 16 times 60 gives me 960. 16 times 60, so that's 960. Okay, so 6 divided by 200 divided by 960. If you can hear, I'm trying it on, I'm doing it myself, and I've got the wrong answer. I see what I've done. Okay, so I actually pressed the wrong button on my calculator. Isn't that good? So 6 divided by 200, that gives me 0.03. Times that by minutes, and that gives me 28.8 minutes, or you can round it up to 29. That's fine. So you get 2 marks for that. So some basic math skills there.